Ever wondered what magic lies behind a centrifuge in a laboratory? How does this spinning marvel separate tiny particles in a flash? Well, today we're about to spin the mystery of centrifuges and dive into their principles, types of rotors, and their wide-ranging applications. So let's get started. First things first, what exactly is a centrifuge and how does it work? A centrifuge is a laboratory device that spins at high speeds to separate particles suspended in a liquid. It operates on the principle of centrifugal force, which is the force that pulls objects outward as they spin. Picture this. When a sample spins in the centrifuge, the heavier particles, like cells or debris, move outward and settle at the bottom, while the lighter, dissolved materials remain closer to the top. This separation is essential for various research and diagnostic purposes. Now, let's talk about the two main types of rotors used in centrifuges: fixed-angle rotors and horizontal or swinging bucket rotors. First up is the fixed-angle rotor. As the name suggests, the sample tubes are held at a fixed angle, usually around 45 degrees. This design means particles move along a shorter distance before they settle, making this rotor ideal for rapid separations. You'll often see fixed-angle rotors used in microcentrifuges and benchtop centrifuges in many labs for tasks like DNA precipitation or protein purification. Next is the horizontal or swinging bucket rotor. Here, tubes start in a vertical position and swing out to become horizontal as the centrifuge speeds up. This design allows particles to separate more evenly and is commonly used in blood sample analysis, where we need clearer layers of plasma and cells. It's ideal for tasks where high-precision separation is needed. Starting with microcentrifuges, these are compact and mainly used for small volume samples, typically in molecular biology labs. Then, we have benchtop centrifuges, a step up in size and power. They're versatile, handling larger samples and offering a range of speeds. Next are ultracentrifuges. These beasts are designed for high-speed separations, ideal for separating biomolecules like DNA, RNA, and proteins based on density. The ultracentrifuge can achieve speeds up to 100,000 RPM, making it essential for high-precision work in genetic and biochemical research. And finally, the clinical centrifuges. These are specifically used in medical labs to separate blood components. They operate at lower speeds, but are incredibly efficient for routine diagnostic tests. Now that we understand the types, let's explore where centrifuges are used across laboratories. In clinical laboratories, centrifuges are invaluable. They separate blood components into red cells, white cells, plasma, and platelets. This process is crucial for blood tests and transfusions. In genetic research, centrifuges are used for DNA extraction and protein purification. By separating different cellular components, scientists can isolate the specific molecules they need for study. In the pharmaceutical industry, centrifuges play a huge role in vaccine development. They're used to purify viruses or cells, ensuring that vaccines are safe and effective. In food science, centrifuges are used to analyze fat content, remove impurities, and ensure food safety. From milk to olive oil, centrifuges help maintain the quality of food products. Finally, a quick note on safety. Centrifuges operate at high speeds, so it's essential to balance the samples evenly to prevent vibration and potential accidents. Always ensure that tubes are loaded symmetrically and never open the lid until the rotor has completely stopped. Modern centrifuges come with safety locks, but it's good practice to stay cautious. So there you have it, the fascinating world of centrifuges. Whether you're working with tiny samples of DNA or large clinical tests, centrifuges are indispensable in laboratories worldwide. Thanks for watching. If you enjoyed this deep jump into lab equipment, give us a thumbs up, hit that subscribe button, and don't forget to ring the bell for more lab essentials. See you in the next video, where we continue to express the secrets of the lab.